Can stress really kill brain cells? Yes, chronic stress can indeed kill brain cells, a phenomenon that has been demonstrated through extensive scientific research spanning decades. This occurs primarily through the action of cortisol, the body's primary stress hormone, which becomes neurotoxic when levels remain elevated for prolonged periods. When we experience stress, our hypothalamic pituitary adrenal (HPA) axis releases cortisol. While cortisol serves important protective functions during acute stress, enhancing memory and alertness for fight-or-flight responses, chronic elevation becomes destructive. Research shows that persistently elevated cortisol levels in the brain can become toxic to neurons. Chronic stress kills brain cells through several interconnected mechanisms: apoptosis, programmed cell death. Studies demonstrate that chronic unpredictable stress promotes neuronal apoptosis in the cerebral cortex, with neurons showing positive staining for caspase-3, a key apoptosis marker. These apoptotic neurons are primarily glutamatergic pyramidal cells located in layer V of the cortex. Excitotoxicity. Elevated cortisol increases glutamate activity, which can become excitotoxic when prolonged, leading to neuronal damage and death. Inflammatory cascade. Chronic stress triggers the release of pro-inflammatory cytokines like interleukin-1b, which directly contributes to neuronal apoptosis. Oxidative stress. Research indicates that chronic stress induces oxidative stress, triggering a catastrophic cascade involving mitochondrial dysfunction, neuronal death, neuroinflammation, and neurodegeneration. Brain regions. Most vulnerable to stress: hippocampus. The hippocampus, crucial for memory formation and stress regulation, is particularly vulnerable to chronic stress. Studies consistently show that chronic stress causes hippocampal volume reduction of approximately 10 to 15%. This occurs through dendritic atrophy and reduced spine density, suppressed neurogenesis in the dentate gyrus, direct neuronal loss in CA3/CA2/CA1 subfields prefrontal cortex. Chronic stress causes the prefrontal cortex, responsible for decision-making, concentration, and emotional regulation, to physically shrink. Research shows that cortisol can literally cause the brain to shrink in size, resulting in the loss of synaptic connections between neurons. Cortical region studies reveal that chronic stress increases the number of caspase-3 positive neurons throughout the cerebral cortex, indicating widespread apoptotic cell death. This cortical damage contributes to cognitive deficits and mood disorders associated with chronic stress. The stress response paradox. Recent groundbreaking research from UC Berkeley has revealed that it's not just the presence of stress that kills neurons, but the body's inability to turn off the stress response. As lead researcher Michael Rape explains, "We always thought that protein clumps directly kill neurons. Yet, we now found that aggregates prevent silencing of a stress response that cells originally mount to cope with bad proteins. The stress response is always on, and that's what kills the cells." Evidence for reversibility and protection recovery is possible. The good news is that some stress-induced brain damage can be reversed. Studies show that neurons have an extraordinary ability to survive and recover from stress-induced damage. Research demonstrates that spontaneous recovery after chronic stress can restore spatial memory function, glutamatergic transmission in the hippocampus, behavioral deficits, protective interventions. Several evidence-based approaches can protect against or reverse stress-induced brain damage. Antidepressants. SSRIs have been shown to protect the brain from stress-induced damage by promoting neurogenesis and reducing hippocampal volume loss. These medications help transform stem cells into adult hippocampal cells, replacing damaged neurons. Mindfulness and meditation. Research demonstrates that mindfulness-based stress reduction, MBSR, can increase cortical thickness, improve emotional regulation and brain structure, enhance stress resilience, protect against age-related brain decline. Physical exercise. Studies show that aerobic fitness and cardiopulmonary function are associated with increased hippocampal volume. Clinical implications. Human studies research in humans confirms the animal findings. Studies show that people with chronic stress exhibit smaller hippocampal volumes correlating with memory deficits, reduced gray matter in prefrontal cortex regions, microstructural white matter changes, cognitive impairments in memory and executive function. Long-term consequences. Chronic stress-induced brain cell death has serious long-term implications: increased vulnerability to neurodegenerative diseases like Alzheimer's, higher risk of mood disorders and depression, cognitive decline and memory problems, reduced brain resilience to future stressors. Conclusion. Scientific evidence overwhelmingly confirms that chronic stress can indeed kill brain cells through multiple biological pathways. The hippocampus and prefrontal cortex are particularly vulnerable, with consequences including memory problems, cognitive decline, and increased disease risk. However, the brain retains remarkable capacity for recovery, and interventions like meditation, exercise, and appropriate medical treatment can protect against and even reverse some stress-induced damage. Understanding these mechanisms emphasizes the critical importance of effective stress management for long-term brain health.